Good morning students. Today we will discuss another topic in the molecular biology, that is especially translation. Okay. Already we have discussed the topic transcription. What is occurs in the transcription? Let us simple recapitulation about the transcription process. Right ma'am? In the molecular biology, you need to gene expressions, especially how the gene two dynamic function. The first one is this DNA replication and second one is the protein synthesis. Here the gene expressions, that is transcription and translation. Translation, right ma'am? In last class we discussed the transcription. Transcription means here DNA to synthesis of mRNA. Okay. The genetic material DNA consists of the information. This information is coded by this and to formulate the mRNA. This transcription occurs in prokaryotes in cytoplasm, where in case of eukaryotes, this transcription occurs in the nucleus, right? And last we have already discussed that in prokaryotes, in case of these prokaryotes, this transcription occurs with the help of only single mRNA polymerase 1. Polymerase 1. Okay, but here in eukaryotes, this is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3 type of essential enzymes are needed to go the process to completion. In these also have the 3 steps. Here also we same the 3 steps. What are they now? Those are initiation, elongation, termination. Elongation, termination, right? Okay, and in this, the termination means ending of the process. Ending of the process. Okay, right. Next here, one other important point in the transcription, that is, here the essential elements, those are manganese and magnesium. Divalent elements, manganese and magnesium and manganese. These are divalent important elements which are helpful in the process, which are going on the complete manner. Right now, and today we will discuss about the translation. What is meant by translation? In central dogma of molecular biology, DNA to mRNA, mRNA to protein synthesis is known as central dogma of molecular biology. But in DNA to simple formation of mRNA at the cytoplasm or in eukaryotes, that is in the occurrence in nucleolus, this process is known as here transcription. But after completion of this transcription, here the translation, right? In the translation, here the transcription and mRNA to protein synthesis and new protein formation. This process is known as translation. Translation, right? Today we will discuss about this topic, translation, both in prokaryotes and eukaryotes. Right now, what is meant by translation? During translation, the genetic information present in the mRNA, the genetic information present in the mRNA, and this directs the order, the deals to the order of the specific amino acids to form a polypeptide chain. To form a polypeptide chain, which means what? Or the polypeptide chains are here nothing but where consist of a numerous amino acid. Amino acids are the structural functional unit of the proteins. Right? That is the definition of translation. During the translation, the genetic information present in mRNA is directs the order of a specific amino acid. It directs a specific order to the specific amino acids and to form a polypeptide amino acids. Those nothing but the proteins. Okay, ma'am. This translation involves these following steps. Those are the first one is activation of amino acids. Activation of amino acids. And second one, the active of or recharging or charging of tRNA, activation of tRNA, charging of tRNA, and next here initiation, right now initiation of polypeptide chain, and next one is elongation of polypeptide chain, and last one the chain termination. These important steps are followed or involved in the process of translation. Okay, ma'am. Total how many? 5. 5 important steps are involved in the translation. Right? Here, let us discuss the first step: activation of amino acids. The activation of amino acids means, right, where a specific, for example, tRNA is there, ma'am. This tRNA is transfers the genetic information from the mRNA to the coding strands, right? And the DNA template is there, ma'am. Okay, dsDNA, double-stranded DNA. Here 3' to 5' end and 5' to 3' end, right, ma'am? Here this is the coding strand. And where this is the template strand, right? Here, this newly synthesized mRNA is closely related to this one. That means 5' to 3'. Right now? Okay. Here the tick box, and here also be another one to add, and the complete process is over. Let us discuss here. The tRNA is specific for a specific amino acids. tRNA is a specific for a specific amino acid. Right. For example, these are written as an okay, tRNA alone, which means alanine. The amino acid is here alanine, where the tRNA val, which means valine. Okay, valine amino acid. Okay, it is a protein. Alanine, valine, leucine, lysine, the essential amino acids. Right. How the activation of amino acids occurs? Let us see. Here, in the amino acids pool, those are activated by ATP. These are activated by ATP with the help of an enzyme. The enzyme name is here amino acid synthetase. In the amino acids pool. Okay. So many amino acids is there, and that's why there are specific amino acids when they are getting ready to attach with the mRNA bondings. Like That's why they can have the how to involve the activation. Why means they are activated. Then only they participated in the reaction. Right, that's why here. Those are the important enzyme here, amino acid synthetase. For example, here amino acid. Amino acid. This amino acid, I am just to draw like this one. Amino acid with the help of ATP, adenosine triphosphates, right? This is the ATP with the help of ATP. In the presence of amino acid synthetase, this is the enzyme. This is the enzyme amino acid synthetase. Amino acid synthetase enzyme. Now this amino acid get activated and forming as a total, the formation of, when this enzyme is participated in the reaction with the help of ATP, this amino acid now get the activate. Which means here is the enzyme, this is the amino acid with the phosphates, and next one is that the enzyme. Right, I am drawing these enzymes. The total this one amino acid synthetase, amino acid AMP synthetase complex. Now it get forms as a amino acid AMP synthetase complex, and it releases the inorganic phosphorus molecules. AMP means adenosine monophosphate. Right, ma'am? Now the amino acid get the activation. Let us discuss about the tRNA, charging of tRNA, which means here just I first discussed that a specific tRNA is a specific for a amino acids, which means the tRNA valine and tRNA alanine, tRNA leucine, tRNA lysine, tRNA histidine, so and so amino acids is there, ma. And the amino acid put the essential amino acids is there. So each tRNA is specific for the amino acids. That's why this charging of a tRNA is called transfer of amino acids. Transfers of amino acids to a tRNA is called the charging of tRNA. And the second step is transfer of, right now, transfer of amino acids to the tRNA. Is called as charging of tRNA. Which means if you are just going to write that tRNA, alanine and tRNA, for example, this like one, tRNA, valine, right? These are specific for this tRNA. The specific amino acid is there, and the specific tRNA is present. Oh, that's why the tRNA is now get the charging, right? And next here, and participated the next step, that is initiation. In the initiation process, okay, what occurs? The site where the ribosomes are involving here, in the translation process, protein synthesis is more important. Yes, the up to 50 to 60% of the cytoplasm is consist of numerous ribosomes. Here the participations of the elements are ATP, GTP, and some other important essential enzymes and factors. Here, initiation. The next step, third one is that is initiation. In the initiation of polypeptide chain, right now? Okay. In prokaryotes and eukaryotes, both the initiation factors are present. These help of this tRNA. Here, the activation. Where the activation? How the transfer of special amino acids to the tRNA? Here just I am roughing up this. Okay. And next here another one, that is tRNA structure is there now. Simple in this activation of tRNA, that is especially this is the acceptor arm, and where this tRNA structure, and here the coding variable arm, right? Here, that's why acceptor arm is attaching the specific amino acids with the help of amino acid synthetase. For example, here a specific amino acid is there now. This specific amino acid is come and attached to this acceptor arm, right? This amino acid come and attach with the help of enzyme, and now it get the complex, right? Now the tRNA get the activation, and now the tRNA get charging. Like now, the enzyme name is here amino acid tRNA synthetase. Amino acid tRNA synthetase. This is the enzyme which is helpful. The active amino acids are command attached at the tRNA's acceptor arm. Now the tRNA ready to get to participate in the reaction. Right now, or the process or the mechanism of the production of a polypeptide chain. Isn't it? This is the anticodal region. This is the arm, and this is also the another arm of the tRNA. Now it is totally formed as a completely. When the amino acid command attached with the acceptor arm of a tRNA with the help of amino acid tRNA synthetase, now it is formed as a amino acid tRNA complex. Now it forms as a amino acid tRNA complex. Right now. Now we have the synthetase. Synthetase is an enzyme complex. Now we get the complex. How we can draw a line this complex? Right. Just I draw here. Okay. These are the arms of the tRNA. Here the acceptor region. Here the amino acids, and here the presence of enzyme, amino acid synthetase enzyme. Now it gets a complex of amino acid tRNA complex. That's why the tRNA now get activated. Right now, or the charging. Next process is initiation. In the initiation of polypeptide chains, right? Initiation of polypeptide chains in prokaryotes, okay, are in the same. Eukaryotes and prokaryotes, here presence of the a special type of factors. Those factors are initiation factor 1, initiation factor 2, and initiation factor 3. Sometimes if you like 2 number or like this also. To our convenience, we will write that like initiation factor number 3. Okay, such type of factors are present in the initiation. Okay, the essential compounds required for the initiation. Those are, okay, now the essential requirements. Some compounds are requirement to the initiation process. What are they? The initiation factors are first one. And next one is the ribosomes, the most important one, that is the ribosomes. These are the first one, and second one is the ribosomes, and third one here, that is mRNA template, the most important one, mRNA template. here lost. Another one, that is the amino acyl tRNA. Amino acyl tRNA. Amino acyl tRNA. This is also most important. These are essential requirements in the initiation process. Now, in prokaryote, the initiation factor is, okay ma, including all the events occurs in the step-by-step process. Now we get expressed in a simple manner of diagrammatic representation. Okay. Right ma? First one is involves of the following steps, that is activation of amino acids, which means here the amino acid, this amino acid is combined with the ATP, and next one is with the help of enzyme, that is amino acyl synthetase, next to complete formation of amino, right ma? Amino acyl AMP synthetase complex, right, and forming of it to phosphates. Inorganic phosphates are releasing in the first step. Next step, second step is the charging of the tRNA. Charging of tRNA means here a number of amino acids is there, a specific amino acid is specific for the tRNA, and the tRNA is arm, which the arm is acceptable here. The arm is accepting of a specific amino acids with the help of the amino acyl tRNA synthetase enzyme is helpful to catching of these amino acids at the region of the acceptor arm. Okay, now it totally completely form as an amino acyl tRNA complex. Then the top step is initiation. In the initiation process, both in prokaryotes and eukaryotes, where the some requirements is there, those are initiation factors, ribosome, mRNA template, and amino acyl tRNA. These four important requirements are needed to completing of the initiation process in the translation. Right? In the initiation process, now we are drawing a simple diagram. Okay, the ribosomes. These ribosomes, for example, this is a small unit and this is a large subunit of the ribosome. Okay, ma'am. Right. This is the 30S subunit and this is the 50N subunit. That's why total 70S subunits, which is mentioned here, that is prokaryotes. Prokaryotic ribosome. Right, ma'am? Ribosome. Now clear? In this way, here 30S and 50S. This is a small subunit and this one the larger subunit. In so small subunit, the mRNA template is common actively gendered, and sometimes we are learn before entering to that part. Here the large subunits is consists of, right now. Yes, we are having a 3 special sites which are present in this slide manner. There presents a 3 special sites. The site is A, and the site is P, and the site is E. Okay. Here the mRNA template is goes from this region, right now. And here this is the 5 prime to here the 3 prime are just where the starting, the 5 prime to starting. That's why we are simply coding on like these 3 prime to 5 prime. Okay, why? Because here the amino acids are coming and joined to this. The ribosome is unit. The complete unit is slowly move on the mRNA template. Okay, let us see here. This is the acceptor, or what we say, amino acids or amino acyl acceptor region. Amino acceptor site, region or site. This one polypeptide site, and here E for exit site. Okay, now these three sites are most important to knowing about here. Polypeptide site, and next one is amino acid or amino acids are come and attached at this site. Amino acids are attaching sites or acceptor site, which means A for accepting or attachment. Okay, now or amino acids attachment site. It is also right, attachment site where the new forming of the triplet codons, right now AUG, GUC, okay, some etc. Amino acids come and attached with this site, and here the polypeptide site forming of the site, and next one the exit site. Let us we discuss here the complete process here now. Okay, in this first step, the polypeptide and amino acid says this is a 30S subunit. This 30S subunit is a smaller one, and in the presence of initiation factor of 3, the mRNA template is now moves on this the attached with the 30S subunits. Okay, larger subunit is there and smaller subunit is shows us the forward or up down, right? And next here the initiation factors. Here, which one is most important in this? The first activating of the charging tRNA is now ready to come and join with these polypeptide chain region P site. Why? Because here P site, the first one is in the prokaryotes formulating methionine. Okay, formulating methionine, that's why we are getting right that is F-met. Methionine. Methionine is an important one that is coding an AUG. Okay, then this one write in a simple manner that is F-met. Okay, F-met. This F-met is come and join with this taking of a tRNA, and especially this tRNA is come and join with here the anticodon structures now. This is AUG. The first codon, important amino acid, which is come and join with this site. Only the first attachment of this polypeptide chain. Remaining or later, all amino acids are come and attached only the amino site. Right? Here, this AUG, here also be anticodons of the tRNA. Anticodons of the tRNA. Here the acceptor arm. This acceptor arm is taking the F-met. Right now, F-met. Now it is connected with each other. Okay. Now this process is going on with the help of initiation factor 1, initiation factor 2 or initiation factor 3. Okay. And next, in the elongation process, again we are observed that, okay ma'am, this is the first step. In the first step, the initiation is occurs when the charging tRNA is come and attached with this mRNA template, especially this is come and attached with the P site. And the P site means polypeptide site. The polypeptide site AUG are come and attached here. And this is the most important amino acids, that is methionine, right? And now this site is slowly gets on this, and next in this amino acid pool, another amino acids is there now. Okay, another amino acids is there. For example, here also be another amino acid. Okay, and here also be another amino acid. Okay, in this pool, amino acid pools, so many amino acids is here. The first amino acid is when going on this process. When going on this process, AUG, F, methionine. Okay, this methionine is combined joined with this process. Right? And here AUG, right now. Instead of T, here we mentioned this U. These are the important codes, important codon. Okay, and there is a nonsense codons also be present now. Those are when they are coming to the end of the tail regions or end of the mRNA template. We are understand that the reaction is stopped. Okay, those are U, and UA, so and so. Okay, right. Here the first step. This tRNA is attached with this region, and next one is elongation process. In the elongation process also, elongation process also includes the elongation factors 1, elongation factor 2, and elongation factors 3, and so on so. Okay, ma'am. And also here GTP and GAT so are essential for in this reactions. The energy is required. Okay, that's why here next step is follows here. The tRNA can get attached with this side and it slowly moves. Okay, moves like this. Here another tRNA is combined joined with this A side. Acceptor region. Right one. These are the arms. Right. And this is also the acceptor region. Here another tRNA, and another tRNA. They carry the special type of